Good afternoon and a very warm welcome to the Chintan Shivir on academia and industry collaboration in medical and healthcare engineering. This session is going to be divided into two parts. In the first part, we will have three eminent speakers who will talk about their work in the context of academia and industry collaboration. And following that, there will be a coffee break of about half an hour. And this will be followed by a panel discussion on healthcare, academia and industry collaboration, and where the three speakers will be joined by other experts on the panel. There will be time given for Q&A, both directed at the speakers as well as the panelists. So without further ado, let me start by introducing our first speaker. Dr. Suman Chakrabarti is Institute Chair, Professor and Sir J.C. Bose National Fellow as bestowed by the Department of Science and Technology, Government of India at IIT Kharagpur. He has won many national and international accolades, such as the TWAS Awards bestowed by the World Academy of Science in the domain of engineering and computer science, the coveted Infosys Prize in the category of engineering and computer science, and the Shanti Swarup Bhatnagar Prize in the year 2013. He is a visiting professor at various leading universities abroad. He has a large volume of impactful publications in top international journals numbering over 550, with citations numbering over 17,000, as well as patents and licensed technologies. He has unique expertise in technology development for the underserved population and community healthcare. Dr. Chakrabarti.
Thanks very much, and it is indeed a great pleasure and honor to be a part of this session, Chintan Shivir. I would like to thank the organizers of Inventive, IIT Madras, and the Ministry of Education, Government of India for facilitating such brainstorming discussion. I can understand that this particular session has a focus, and as the beginning speaker, I thought that I would like to stick to the focus rather than talking about more general aspects of technology for healthcare. And as I understand that the main focus phrase for this entire panel is industry-academia collaboration with particular reference to healthcare, and that also in particular in the Indian perspective. And that is how I have tried to organize my talk. Now, when we think of the Indian perspective, we of course have to be bothered about not just one or two isolated locations, but the entire country where we have a natural diversity. And the diversity effectively means that we have certain locations where you have very high level facilities available, and there are locations where hardly facilities are available. There are people who are in need of the facilities, very urgent need, and the need to availability ratio is just the opposite. Now, on the top of that, when we think of the healthcare delivery at resource limited settings, be it rural or be it urban, there are additional constraints which come from affordability along with accessibility, and that means that we need solutions for these problems in a way which may be different from many other countries who are working on similar topics. AI has come in a very big way, and with AI, we are in a position to give solutions, or rather propose certain solutions which combine technologies with clinical expertise, medical knowledge, and diagnostic reasoning, all combined together. Having said that, there are still several issues, and those issues are certain factors which will usually be possible to address if we have a very strong industry academia collaboration in healthcare. Now, of course, industry academia collaboration is well known in every area, and typically in areas where technology plays the forefront. In healthcare, technology acts in the forefront only very silently or subtly, because in healthcare, the healthcare professionals come in the interface more emphatically. So, we have a rising disease burden, both communicable as well as non-communicable diseases. Non-communicable diseases are almost like epidemic, like diabetes and complications related to that. So, that is one side. We have extreme regulatory challenges, and regulatory challenges are there which we have to navigate through, but at the same time it is impossible to work in healthcare technology ignoring the regulatory aspects. So, we have to see what best can be done through industry academia collaboration. And global competitiveness, India has the potential to lead in medtech and biopharma because of several reasons, like the Indian scenario which drives innovations for the solution will be in a position or is already in a position to come up with certain solutions which are really unthinkable in terms of like the way in which people think in highly resourced labs. So, there are key areas in which industry academia collaboration can take place in the broad healthcare scenario. Medical devices and diagnostics is perhaps one of the most common in which the IITs work in the context of this program. Pharmaceuticals and drug discovery, of course, there are several very reputed national institutes. IITs are not known to be in that league for at least the drug discovery and pharmaceuticals purpose. AI and digital health is again an area where IITs are very strong. Clinical trials and biotech innovation, and clinical trials is I think going to be one of the forefronts of what is going to be thought about in the coming months or years. And public health and preventive care, which is very, very important, and currently there is a, what I would say, emphasis more like a charity mode rather than a professional industrial mode. So, sooner we get out of this charity mode for doing good for the poor community and get into a professional industry academia mode, I think the quality and impact will be more. So, we talk about industry academia collaboration, but we recognize that it is not just industry academia, it is that very famous triple helix model where government comes in a very big way, and these are connected by funding and strategic needs, new products and concepts, and there are key benefits that the government derives along with of course the human resource and the scientific development, that is employment generation, taxes and benefits to the citizens, and this is very, very important. But having said that, every activity has a challenge, and for the interest of time I would browse through the main pointers which I think as a challenge. The regulatory and bureaucratic hurdles I have already mentioned. Sometimes they have quite a long process, and in an emergency situation like COVID, something could be done, but everything is not like COVID for which similar things are being done. So, there has to be a sort of a simplification which should come from some the responsible source. Mismatch in objective and expectation, this is something as a teacher I have always felt that academia focuses on fundamental research, publications and long-term knowledge creation, and even academicians are recognized for these mostly, although people will talk about many other things, but any academician going for a promotion interview primarily how many PhD guidance and how many papers and all those things are usually asked. So, but industry prioritizes market-driven solutions, quick commercialization and return of investment, and that cannot be disregarded or dishonored. So, there has to be a way in which this needs to be bridged. There are funding issues, with a limited government funding, and government funding is quite good, I would imagine, but it has its own limitations. Industry hesitation is there, and lack of venture capital support for many of these activities. Another point which I feel very concerning is weak IP framework, and because of this weak IP framework, there are and clear lack of sort of even nationwide policies, there are localized policies, and therefore I see often when a product gets developed or a technology gets developed and gets towards the commercialization phase, some disputes evolve, and that can slow down things. And also unfortunately that academia produces highly educated people, but in many cases there is a shortage of industry ready people or industry ready human resources, and that has to be looked into from possibly curriculum and more project embedded activities in the curriculum. And my own impression is that biomedical engineering, which now is there in many places as even undergraduate courses, it is like it is still acting like a mixture of many things, but with no core expertise being developed, and that is not always helping. And in terms of infrastructure, there is limited infrastructure and limited research facility in many academic institutions, and industry often needs to invest, and where industry will invest, whether industry will invest in the research park or on the campus or at their own place, this is also something which needs to be sort of deliberated on a case-to-case basis. Data privacy and security concerns, typically when lot of patient data and other medical data is being worked with for medtech development and research. There are cultural and structural barriers, like as I told that professors have a cultural way of responding to a particular need, and industrial people, and there is low awareness and engagement from industry, and many industries are actually not that much aware of the immense potential that will be there in many of the university campuses. And in medtech, there are additional a bit of more technical issues like challenges in scaling innovations. So scaling means not just business scaling, it is also scaling in terms of technology, whatever is developed in the lab. Many times we find that for the end dissemination, the product has to be completely re-engineered, and it is not always a good thing. And possibly if we start with industry academia collaboration from the very beginning, the product will get, or the technology will get that foresight that how it needs to be done. So but if it is done, there are lots of opportunities and benefits. Accelerated research and innovation, it can definitely accelerate what R&D is being done at both the places. Cost effective and scalable healthcare solutions, enhancing clinical trials and drug development. This is very, very important, and I think with this collaboration it can only grow. Workforce development and skill development, and this is where academic institutions are good, and this is where I think that can be leveraged upon, and these kinds of activities can be embedded within the curriculum, that more industry ready healthcare professionals emerge. Fostering AI and digital health innovations, this is already there, but with a caution that I always see extremists, like people who are for AI saying AI can give all solution, and people who are traditionalists say that this is the core technology, AI is of no use. So that balanced view of merging AI with the core technology is very important. And with all these, improving rural and preventive healthcare, this is one of the India's major problem, and industry academia activity in this particular area is quite inadequate. Policy and regulatory reforms very much needed. Streamline approval processes, be it regulatory approval. So typically regulatory approval has a sequel, but if it could all be through a single window process, that could streamline. Clear cut IP policies in this particular area. Well, different institutions have this, but there is some sort of mismatch. And government led incentives and grants, like give tax incentives, subsidies, innovation grants, what government gives, like for example BIRAC's big grant. I am one of the a lot of beneficiaries of such a grant. So I recognize that it's a huge boost for anyone, be a professor, be a student, but I think there should be more. Strengthening university industry collaboration models, I would say co-development of technologies is very important, and the co-development assures that industry quality things are at least being kept in mind even at the lab stage when the research is being done. University embedded innovation hubs, and this more and more should be there. IIT Madras is already very good in it, but I think there are several other institutions should follow. Private public partnership for healthcare infrastructure, so that may help upgrading the university research facilities, because government alone cannot do all these things. Co-design curriculum and training program, I think this is very, very important, because India's one of the strong points is human resource, and this kind of domain where we sort of need continuous upgradation of knowledge and expertise hands-on, I think there should be more co-designed curriculum and training program which is jointly participated on by the industry and academia, and industry sabbaticals for professors and research internships for students. Very, very important, where we can have this. So it will of course be connected with several other very common factors which are there not just in healthcare but in other areas like AI and big data research, virtual R&D collaboration platforms like creating, for example, DST, CII technology gateway is already there. If more such virtual gateways are there, people will have a single window access to most of the activities which are going on. Stronger incubators and accelerators, and in some way how to stimulate the venture capitalist and industry funding. So I would like to wind up in the next 2 minutes with a bit of input on, in this respect, what we have been doing at IIT Kharagpur in our research group. So fortunately we have a project which is funded by the DSIR on what we call as CRTDH, Common Research and Technology Development Hub. It is like you can say industry academia bridge, but more focused on MSMEs. And the whole idea is that this particular hub is on affordable healthcare, for which I am the PI. And initially we started with developing technologies and transferring the technologies to industry, the standard mode. But then we realized that there is it's a one-way traffic. But the MSME, startups, industries, they also have certain expectations, and we therefore got into a sort of a collaborative mode, and which I found it's more engaging, because in that way we have developed prototyping and testing facilities, open source instrumentation, which anyone can use with a subscription model. So we have introduced a gold, silver, platinum membership, which anyone can take, and then that company or individual will have seamless access to all these things which are being done. IP protection will be there, but that cannot sort of prevent from this cross talk. Market access opportunities, support for scalability, sustainability, shared lab access for incubation, and sustainable revenue model for such incentives. So the last one that I will go through is my own experience of such a lab to market journey. So frankly speaking, when I started my own research on say microfluidics for healthcare technology, possibly around 15 years back or so, I never had this vision. So we developed technologies in the lab, and one such technology which at that time was not so common was like doing a large number of blood tests on a paper strip with a single drop of finger prick blood. So we had a device in which we had that embedded sensors, camera and other electronics, and you take that finger prick blood and the strip you insert into the device and get the camera view and do the image analysis and get the prediction of be it blood glucose, hemoglobin and so on. Now when we transferred it to one such industry and we were interacting, we realized that we have so many glucometers, or several such meters, many such areas we do not have such devices. But be it a device there or not, the challenge is that say someone who is having to monitor something throughout the day, Now the person has to carry that meter and that auxiliary thing, and it creates a lot of inconvenience. So then we realize that the challenge why we cannot get rid of the device is that the device gives a quality control of the ambience in which you take the image, or maybe you do some measurements. It creates a controlled environment. Now nowadays with the advancement of smartphone, you can do certain things without control, and this also could be done. But that lack of control or the variability from one smartphone to the other, one user to the other, one ambient condition to the other will give erratic output, and that needs to be rationalized, and that required an innovation in the algorithm, and that is what actually with this startup we did, Smart QR Technologies from Pune, and together we developed this technology which this entire brand was sort of incubated at IIT Kharagpur. So it's a Smart QR brand, where smart is for smartphone and QR is for QR coded. So it's basically a strip, QR coded strip, in which you can put one drop of fluid and then get test done. So currently there are so many in the pipeline. 2 are already CDSCO approved and they are in the market. And I would just give an example with some demo video of these 2 before we finish. One is HemoQR. So this is a hemoglobin test. So what you can do is that you can carry that one packet in which you have the lancet needle and the paper strip. At any point of time, you just take that finger prick blood and put it on the strip and take the smartphone image. Any smartphone will do, and in the Google Play Store the app is already there, and then the app will display the result. You do not need a device. So, and this is something which is gaining quite popularity. Another test which from my side it is a bit of a personal satisfaction. We try to work a lot on women's health, and typically women's health at underprivileged locations. So this hemoglobin test is one such thing, because anemia is a major problem for women in resource limited settings. And also there are many women, like this is a case study which we did at Sundarbans. Sundarbans are very what I would say as geographically interesting, but I mean in many ways there are lots of challenges that women face there, and one such challenge is a recurring infection, vaginal or several other types of infection, for which they cannot really approach any care provider, because simply there is no trained gynecologist available, and the local workers who are there, the so-called frontline workers, there is a cultural barrier between them and the women who are suffering. So we have this PrEP QR test, which is of course commercially available. So this is like a cultural transformation where the woman is trained to collect the self through that cotton swab, the self collection of the swab, put it on the paper strip. It measures the vaginal pH very accurately, up to 1 decimal with 100% accuracy, quite validated. And with other supporting evidences and all, we are trying to give a screening to them. In a way, they don't have to visit the doctor. They have to just give certain answers, and they can tell it verbally also, and then it can be recorded. It is passed on to the primary care provider who is at a resourced place, and in that way, at least our field work at Sundarbans, I found that it is like a what I would say a life transforming lesson for me as a developer of the technology. I have never imagined that this could be done. So such kinds of things, these are the opportunities in the Indian context. And typically industry of course is always in the high end activities and not many times focusing on these kinds of needs, simply because these are not sort of industrially viable, reachable locations. So if now this somehow could be bridged, like for example I have given this IIT Kharagpur model. So we have this the R&D units for medtech and this in dissemination model. So we have a back and forth like interaction, and this is where industry can chip in and can do many things in addition to just healthcare, like training of the rural women and youth, and it can therefore give rise to a huge convergence with what can be done for the benefit of the common people. And in that way, industry-academia collaboration beyond technology in the healthcare domain, of course, can do good things with science, technology, exciting things, but more importantly, can emerge as friends of the community. So, in that way, we can strategize. I'm not, getting into the details, but, I have a sort of a vision that we can have some immediate steps next 1 to 2 years, some mid-term strategy, which is more like, it is going to give a longer term benefit, and a long-term vision, which we can set up for the road ahead. It of course will require policy reforms, strong incentives, investment in skill development, and greater industry stroke academia awareness. And with all these things, I think that there is such an exciting opportunity simply because we have challenge. And if we can convert that challenge into opportunity, I think that together academia and industry in India, we are of course going to be at the forefront, and I would say make our Honorable Prime Minister's vision of Vikshit Bharat and Make in India, all these so realistic. Thank you very much.
That was an interesting presentation. Thank you, Dr. Chakrabarti. Dr. Chakrabarti has indicated that he will be leaving after the coffee break, so he has given us a complete bird's eye view of academia-industry collaboration in the context of the work that he's giving. And the audience can ask questions at the end of this and network with him during the coffee break. I'd like to call upon our next speaker, Dr. T.S. Suryanarayanan, Director of Winstrom. He's a mycologist, a World Academy of Sciences or TWA's visiting professor, Fulbright Nehru Senior Fellow, Honorary Research Associate, University of New Brunswick, St. John, Canada, Research Supervisor, University of Munster, Germany. He's ranked in the list of top 2% of researchers across the world in a study done by Stanford University scientists. He has over 150 publications, over 7,000 citations, an H index of 47. He has remained the President, Mycological Society of India in the year 2016-17.
Thank you. At the very beginning, I would like to thank IIT Madras and Dr. Narayanan, Inventive, for giving me this opportunity. So let me straight away go to my topic. So I'm a mycologist. My lab specializes in the study of fungi. So just like our gut houses diverse microorganisms which provide us with positive and negative effects, the plant leaves and roots also have native microorganisms inside them. And these are called the endophytes. Endo means within, phyte, plant. They are within the plants. They don't cause disease. They are symbiotic. And they actually protect the plants from insect and pathogen attack, and also improve the tolerance of the plants to certain abiotic stresses like salinity and dryness. Recent studies show that such endophytes, especially, the endophytes within plants are fungi, bacteria, actinomycetes, but the fungal endophytes, they've shown that they can actually be used to improve the tolerance of crops to climate change effects. So, additionally, endophytes, when I say endophytes, I'm referring to fungal endophytes. They have unique genes which are not seen in the non-endophytic fungi. And so some of their traits are significant or of significant technological use. So my lab has been studying this fungal endophytes of plants for the past 40 years, and we have now in our lab perhaps the world's largest collection of these endophytic fungi isolated from plants of different parts of India. We also isolated from forest trees of the Western Ghats, from lichens, from inside seaweeds, also from inside marine sponges, salterns, mangroves, and from different parts of the country. Okay, so, well, we are basically mycologists, so our specialization lies in isolating these fungi, culturing them, getting azonic or pure cultures of them, identifying them, and then doing some very basic work to see if they produce some novel metabolites or catalysts like enzymes. But there it stops. Our specialty cannot take us beyond this. So we had to collaborate with different labs in India and also outside India, and in projects funded by DBT, SCRB, Ministry of Environment, World Bank. And based on these studies, we found out that some of our fungal cultures produce some interesting metabolites which could be technologically exploited. Yes, so we'll go and see what are these things. So our research has shown that endophytes are an untapped source of water-soluble melanin pigment, anti-malarial metabolites, novel antibiotics for managing antibiotic-resistant pathogens, novel biomass-degrading enzymes, novel chitinases and chitosanase enzymes, pharmaceutical enzymes, anti-cancer metabolites, anti-coronavirus, SARS-CoV-2 metabolites. Then they can be used for improvement of crop production under climate change scenario, and a few other enzymes like carrageenase. So I will quickly briefly explain these results, and then we can see how this. For example, water-soluble melanin pigment. Melanin is it is called eumelanin, is a pigment that is found in humans, including our hair, but it's not water-soluble. So if we can get water-soluble melanin, then it makes it easy to extract. And the water-soluble melanin is called pyomelanin, and pyomelanin is easily extractable since it is water-soluble. It screens UV light, and it is not photodegradable. It can be used in space travel as radiation shield. It shows antimicrobial, antioxidant, anti-inflammatory activities, highly thermostable, and has properties that are promising for application in bioelectronics and energy. No wonder it costs about 80,000 rupees per gram in the international market, this pyomelanin. But so far, only bacteria have been studied for the production of this water-soluble melanin. And we have shown that fungi, especially endophytes from isolated from roots of mangroves of Sundarban, they produce this chemical, and also from Pondicherry, they produce this water-soluble melanin pigment. Okay. Then, in collaboration with ICGEB, we showed that some of the endophytes which are present in the tree leaves, barks, and seaweeds are a potential source of metabolites which inhibit drug-resistant malarial parasite. Further, our data suggests that antiplasmodial potential is more widespread across diverse fungal genera. Right. So, novel antibiotics for managing antibiotic-resistant pathogens. Once again, I want to stress that these are just basic work. We have not identified the metabolite. It's a crude extract, the secreted metabolites of the fungi that we are looking at. So, if somebody takes it up, there is a 90% chance that it is probably an already known antibiotic or an antibiotic which works but also is not good for normal cells. But unless we do the work, we wouldn't know. Well, for example, we gave some of our fungal extracts to ICMR here in Chennai TB Research Institute, and they found that a couple of fungi produce metabolites which inhibited clinical isolates which were resistant to isoniazid and rifampicin, TB bacterium, tuberculosis. But there it stopped. Because in India we have this major problem. If somebody is doing some work, whoever it is, if the person reaches 60 or 65 years, the person may not die, but the work dies. That's the end of it. That Dr. Saroj, if I remember her name was Saroja, she was interested in doing this work. She was in this ICMR. After doing this work, she retired. There it stopped. Okay. And then, yeah, so here in collaboration with the Jawaharlal Nehru Research Center in Bangalore, we did this work. We isolated some fungi from solar saltan. So they were highly salt tolerant. And they produced metabolites which inhibited human pathogenic Candida species. Human pathogenic Candida are they are there, but they are clinically not very dangerous. But when in immunosuppressed patients, it becomes a very major problem, and it's very difficult to control this Candida infections, especially if it is caused by Candida tropicalis and Candida auris, because these have become resistant to all the 4 known antifungal antibiotics. But some of these fungi produce some metabolites which inhibited this drug-resistant Candida, those which were resistant to fluconazole and capsofungin. Well. Okay, so we are in collaboration, we do some research with Shankar Nethralaya Eye Hospital here in Chennai, and Dr. Anand there, he tested some of our extracts in the basic work, and he found that some of the fungi were very effective in inhibiting Staphylococcus bacillus cereus, Streptococcus pneumoniae, and Candida albicans, which causes lens infection, iris infection in immunocompromised patients. Yeah. Oh, well, now this has nothing to do with health directly, but a conversion of biomass to biofuel. This is the best method they use is the biomass is taken, and then it has to be first treated with high temperature and acid to break the covering called lignin. Only then the cellulose is exposed, and then it can be converted to sugar and finally to alcohol, ethanol, or whatever. But for that, it becomes tough because I'll tell you that a little later. Here, we found that some of these fungi, which we isolated from sea grasses and seaweeds, they produced salt-tolerant and salt-induced xylanases and xylosidases enzymes. So we concluded that seagrass and seaweed, dead seaweed and seagrass, can be used as for solid-state fermentation medium for the fungi, thereby reducing the cost of production of biofuel, because you don't have to use, now they use corn cob, rice bran, sunflower head as substrate for growing these fungi. Instead, you can use this dead seaweeds and seagrass. And so salt-tolerant saccharification enzymes like xylanases could play a crucial role in deconstruction of biomass, especially when ionic liquids are used for pre-treatment of LCB, or lignocellulose biomass. So that's another interesting thing. And then, as I told you, when we take plant biomass and convert it to biofuel like ethanol, the first thing you have to do is treat it with high temperature and acid so that the lignin is sheared, it is broken open, so that the organism, the bacteria, the enzyme cellulase can be used to break cellulose into simple sugars, and then the yeast can be used to convert sugar into ethanol. Now, when this pre-treatment, when it is done to shear the lignin, when you use high temperature and acid, the lignin breaks. At the same time, it produces, because of the reaction, it produces some metabolites which inhibit the downstream process. So you have to wash. After doing this, you have to wash it. So you waste a lot of water and energy. So the cost of biofuel production increases. If you can find a cellulase enzyme which can work in the presence of these pre-treatment inhibitors, then technically or theoretically, it can bring down the cost of biofuel production. We found some of our fungi, they produce these enzymes even in the presence of several inhibitors like furaldehydes, phenolics, and tannins. And not only that, some of our fungi use the inhibitors as carbon source. So they grow, they reduce the inhibitor, so you don't have to wash it. And then they also produce cellulases which are not affected by the inhibitors. So that step of washing the pre-treated lignocellulosic biomass with water can be skipped. So you can save a lot of money. Okay. So we found that these fungi produce endo and exoglucanases which function in the presence of derived phenolics. Some are active in the presence of tannic acid inhibitors. Some of the fungi produce thermostable endonuclease enzyme. Some fungi utilize the inhibitor as carbon source. And we in collaboration with VTT in Finland, we also tested if these actually work in the presence of the microcrystalline cellulose substrate which is used in industry, and they worked very well. In fact, higher yields of sugar were obtained in the hydrolysis of cellulose when phenolics were added. This was surprising. Phenolic is an inhibitor, but the fungus could use it, and then it produced the enzyme. These are the papers that we published regarding these things in different journals. Yeah. Now, chitin modifying enzyme. You know that chitin is broken down by enzyme chitinases, and then the product, which are called chitosanes, are used in for control of microbes and tumors, wound healing, osteoarthritis, wastewater treatment, and drug delivery. Our results showed that our fungi are a good source of different types of chitin modifying enzymes. There is a high genetic diversity in endophytes with reference to chitinolytic enzyme production. The same species of the fungus isolated from different plant species differed in their capacity to produce enzyme, differed in their capacity to produce different isozymes. And so endophytes can generate chitosanes of various lengths based on which their properties also differ. So we can tailor-made chitosane products can be got by using enzymes instead of the acid wash technique, which gives you a lot of different types of chitin, and you have to spend money to separate them based on their molecular weight. So these are the papers that we published based on that. And here is another thing, pharmaceutical enzyme. You know, there is an enzyme called L-asparaginase, which is used for controlling, for managing acute lymphoblastic leukemia in children. So the cancer cells, they do not synthesize this asparagine amino acid. So they take that asparagine from the blood, from the asparagine which is circulating in the blood, which is secreted by normal cells, excess is secreted. So one way to manage this disease is to inject asparaginase enzyme into the blood of the child. So the enzyme will break down the asparagine in the blood. So the blood is deprived of asparagine, and then the cancer cells are starved and they die. It sounds good, but there were two problems. One is the marketed L-asparaginase is obtained from transformed E. coli, and it differs from the asparaginase of the humans by about one, or I don't remember, maybe 2 amino acids. So because of that, allergic reaction can take place. One, it does take place. And two, the enzyme asparaginase is not strictly substrate specific. It also acts on glutamine, and it all breaks glutamine to ammonia and another product. And so that can cause neurotoxicity. So they have to monitor the child continuously. So if you can get a eukaryotic L-asparaginase, which has a similar amino acid sequence as that of ours, and if it is functioning only as asparaginase and not as glutaminase, then that would be a good choice. And we found some of our fungi producing L-asparaginase, functioning only as asparaginase and not as glutaminase. This in collaboration with the IIT Madras here, with Gumadi, Professor Gumadi. Anti-cancer metabolites, in collaboration with our German scientists, we found that, some of the fungi produce metabolites which can inhibit cancer cells. Unfortunately, the one which was very effective found out it was toxic to normal cells also, so we couldn't proceed further. Here is a very simple work, very basic work, which was done by Regional Center of Biotechnology, DBT, in Faridabad. They found that some of our culture filtrates of the fungi inhibited SARS virus, and they said the extract of endophyte 418 had some hope, but then we couldn't proceed further because that student got a job and he. Finally, this is the thing. So if we have shown that if I take a fungal endophyte, just 1 minute, this is the last slide. So if I take a fungal endophyte from a plant which is growing, a weed which is growing in, say, in a salt marsh or in the mangrove surrounding, I take it, and then I find that it is resistant to, it's able to grow in high salt concentration in the medium. So it is adapted because it is coming from a saline infestation. You can take that, grow it in culture, purify it, get the spores of that, inoculate it into your rice plant, which is not able to tolerate salinity. But once this endophyte goes into it, the rice becomes tolerant to salt, saline, and it produces seeds. The endophyte goes to the seed, so you don't have to inoculate it again. From the seed, you can get the second generation that will also be resistant to salt conditions. So similarly, if you take a plant, and if you take an endophyte from a plant which is growing in Rajasthan desert, that they have shown, and it is more resistant to high temperature than the normal thing, and if you can inoculate it into a plant, that crop becomes a little more resistant to heat. So there is a possibility that climate change scenario can be managed by inoculating such select fungal endophytes. This is a very interesting thing because it is non-invasive, it's not genetically modified, and the crop itself has its own fungal endophytes. We are not introducing anything new, we are only introducing a fungus with a different trait. So all we have to do is see that they don't produce any mycotoxin, then it will be of use. So so these are just basic results, that's it. So you just opened a fraction of that treasure box and we found there is something there. But help is needed to open it fully to increase the bioeconomy of the country and the genetic resource of the country. So I'd be very happy if somebody would be interested. We are run by the Ramakrishna Mission, so we are not interested in money. We would be willing to give our cultures free of cost, and so it can work. All we need is some collaboration so that we could see the end of it. Thank you.
Thank you, Dr. Suryanarayanan, for a very comprehensive overview of the potential promise and applications of fungal endophytes. I call our last speaker. Kumar Shankaran is the CEO of Leucine Rich Bio. He is a business leader with a professional experience of over 15 years in healthcare research and delivery. In his current role, he is spearheading the growth and innovation in the microbiome domain for the Asian market. He is passionate about driving the paradigm shift from sick care to healthcare.
Hello, everyone. I'd like to thank IIT Madras and the organizers of Inventive for inviting me here to give this talk. Today, I want to juxtapose our journey as a startup, a decade-old startup working on microbiome, and how probably academia can help bridge a lot of gaps that's there in this domain. Even 2 years ago, when I used to give talks on the microbiome, most of the audience would not know what the microbiome is. And I think in the last 2 years, the awareness of the microbiome has increased tremendously. For those of you who still are new to this domain, if you're craving for a coffee or a snack that's going to come up after this talk, it's most probably a bacteria in your gut signaling to your brain saying, feed me that. So the microbiome is all about the microbes that are, especially when you talk about the human microbiome, it's all about the microbes that are there in and on our body. This is a common definition that people keep giving about the microbiome, the second genome. And one of the reasons this was prevalent for the longest time is the way to measure the gut microbiome especially was by looking at the DNA sequences of the microbes that were present. So the way you do it is you collect fecal samples or poop samples of an individual, isolate the microbial DNA from it, and sequence it. But why does it have to be DNA all the time? I mean, can we look at metabolites and determine what the gut microbiome would look like? Sir was talking about imaging blood spots to come up with blood sugar levels. Can we do a yucky photo of a fecal sample and determine what the gut microbiome would look like? So it does not have to be DNA alone, and these are some of the research that is going on worldwide that I think we should also try and see how we can incorporate over here. Trust me, transporting fecal sample from all across the country is a very, very difficult task. So if this can be done at home, it'll be really helpful for me. So when we talk about the human microbiome, the gut microbiome, gets a lot of importance. But that's not only it. I mean, it is a significant part of it. But there is the oral microbiome, there is the skin microbiome. Now people are saying that the brain is infected with microbes. Sorry, infected is the wrong word. The brain is occupied by microbes, and every region, every organ has its own microbial signature. So there are different parts of the body that still needs to be looked at and seen what the signatures are. We recently got samples of cornea scrapings and to see what the microbiome population of that is. So there's a lot more exploration that needs to be done with different parts of the human body. The gut, of course, is the most influential because 95% of all the microbes are there in the gut, and the way it's very, very important. I mean, the way you feel, the way your health, your disease, a lot of it is actually is controlled by the gut microbiome. And the way we look at the gut microbiome is through a surrogate right now. We're looking at fecal samples to try and understand what's happening in the gut. Can we instead send a pill inside to the gut and see what are the microbes that are there? It's been done in cattle. So why can't we do it in humans? So there's another device that you can think of putting it into the human body to get to know about something that is so important. I mean, I usually put this slide up to tell people it's not a weight loss mechanism. Don't remove all the microbes in your body. But I wanted to put this slide up to discuss 2 points of view. One. There's so much microbes in the gut that fecal transplant, that is taking the fecal sample of a healthy patient and transplanting it to a diseased individual, is actually helping a lot of the individuals worldwide. US did a lot of trials on this. Australia is dominating this in terms of the kind of trials that are happening. It's called fecal microbiota transplant. AIIMS Delhi has started a clinical trial very recently. We'll be starting our own trial on IBS also in the next few months. So there's transplantation of fecal sample, which is helping in a lot of diseases. So much so that we get calls from patients with Parkinson's disease calling up and said, we've got FMT done, and we see a lot of improvement. Is there a way that we can do this in India? So there's a lot of scope in that. The second thing is, with all these microbes, that are there, we can create our own line of probiotics. So all the probiotics that you see in the market, whether it's pharma grade or food grade, all the probiotics are most probably coming from a US culture or a European culture, or the Japanese one is also very, very famous. But most of those strains do not colonize the Indian gut. And India is so diverse. A strain that we can isolate in Northeast India might not work in South India. So why not use your own sample to create your own probiotics? labs in CFTRI have started working on that. There are a few probiotics that will be coming out from actually baby samples shortly. So that's some interesting work that's going on over there. It's like a fingerprint, the gut microbiome, but it's also very transient. It changes over a period of 3 to 6 months time. It depends on your lifestyle. It depends on the kind of food you're eating, where you're eating. I'm sure people working in IIT Madras and who are coming to campus on a regular basis have a slightly better microbiome than people working in the rest of Chennai. That's because of all the greenery that you're surrounded with. I say this with a little bit of confidence because if you give me any sample today, I'll be able to predict whether it's an urban sample or it's a rural sample. But with all this knowledge, we still know only what an unhealthy microbiome looks like. I'm still trying to discover what that healthy microbiome is. Sir was mentioning non-communicable diseases in his talk, and 90% of the non-communicable diseases can be traced back to the microbiome. And for looking at these diseases, I mean, it could be something like rheumatoid arthritis, could be anxiety, could be depression, and all of this, you're getting insights from looking at the gut microbiome. You can even look at the oral microbiome and detect if somebody's progressing towards pancreatic cancer. To look at this, you don't have to measure all the organisms that are there. Can we look at devices that can do just a handful of microbes, and that can be a at-home test? And this was a challenge thrown to us from the global pharma company GSK. They had developed a drug for some of the poorest parts of Africa, it's a poor region. They couldn't just supply drugs to everybody out there. They wanted a simple diagnostic for measuring a certain disease related to the gut microbiome, and they wanted a device that could only do over 3 or 4 microbes. So how do we get point-of-care devices that are looking at these microbes for very, very specific diseases? This is dysbiosis and eubiosis are the terms that now have become very popular. I mean, dysbiosis is when your gut is imbalanced, and eubiosis is when it's supposedly balanced. We know what dysbiosis looks like. For example, if I were to take, especially the students of here, if I were to take your sample, I would see anywhere between 1,500 to about 2,200 different species in your gut. Anybody suffering from a chronic illness, the number of species would have come down to about 800 to about 1,000. So the lesser the diversity, the more problematic it is for you. What if the gut was super diverse? What if we had about 5,000 species in? Does that make you super healthy? We don't know the answer for that. All we know is lesser diversity is a problem. What is that threshold? That is something that still needs to be determined. This is the Hasta tribe. They have about 600 different varieties of food every year. So this is vegetables, fruits, meat, all kinds of food put together, 600 different types. So it's very seasonal. What they eat is very, very seasonal. And they also have the most diverse gut microbiome known to mankind today. Absolutely no cases of reported lifestyle or non-communicable diseases. No diabetes, blood pressure, no cardiovascular diseases, none of that. And the sense that we're getting is it's because of the diversity of the microorganisms that are there. So again, it goes back to that question: what is that diversity ratio that we need to get? Can we keep increasing the diversity and make it better and better for us health-wise, or is there a limit to where we can go? So in our decade-long journey, the one thing that we've been trying to do is trying to establish what the Indian microbiome signature would look like. Back in 2014, when we started this off, I think my sample might have been one of the first samples ever to be, sequenced in India. So that's how far back our journey went, and we went across India collecting samples from all parts of India, and started mapping what the signature of the microbiome would look like. I wish there was an academic paper back in 2014 telling us this is how the Indian microbiome signature looks like, and population microbiome is something which is still the need of the hour. Can we establish a healthy baseline? So the one way we tried to establish healthy baselines was we went after professional athletes. So these are athletes who are representing India at the international level. So it could be cricket players, badminton players. I can't take names of here, but all of them are representing India at the international level. And we thought they would have the best gut microbiome. They're so fit, they're so they take care of their health so much that they'll have the best gut microbiome. Turns out they have probably one of the worst microbiomes. That's because their body is under so much of pressure, both mentally, physically, and given their lifestyle, the travel and the kind of food that they eat, their gut microbiome is really bad. So how do we establish a good, healthy baseline? How do you define healthy? How do we get to a point where you say this is what you need to achieve? We have that for glucose monitoring in terms of HbA1c. Can we get that to a kind of a number for the gut microbiome? Lastly, I know a few universities have started doing the Indian tribal gut microbiome. I think a lot of samples are being collected. The publications are not yet out, but there's so much of diversity within India. North, south, east, west, this varied population, all kinds of food habits. And the one thing that is common across India is all of us traditionally have been eating a lot of fermented foods. Recently, there was an FSSAI booklet that was published on all the fermented foods. I think about 26 fermented foods that were present in India, and one for every region. I'm guessing kanji is the most famous one here in Tamil Nadu. So how does fermented food help, especially when you go into a non-urban setting? How good is it? How is it making? How is that helping us in combating disease? Dr. Suryanarayan was talking about mycology and the amount of fungal species that he's been studying. The one difference between the Indian gut versus a lot of the Western gut is we find a lot more fungal species in our gut. I've seen enough data to know now that there is no good bacteria, bad bacteria, no good organisms, bad organisms. I mean, everything is in relative proportion. So, and I'm sure these fungal species are adding to the diversity and adding to the functional metabolism that's going on. I pulled out data that we've taken in the last 6 months. So Bucksweeks is our kind of a gut microbiome diagnostic kit that we send out. So so the last 6 months we've done close to about 25,000 samples. But the interesting part of this is those 25,000 samples are now beginning to come from outside the main metro cities. It's coming from Tier 2 cities, and we're getting to see a lot more interesting data and a lot more interesting microbes that are coming out of there. We also conducted a lot of clinical trials over the years, trying to establish again what an unhealthy microbiome looks like. But getting data from varied diseases will also help us in understanding what the baseline microbiome, healthy microbiome would look like. And for all you computer nerds out there, the amount of data that we generate is humongous. I mean, we've done in the last 6 months, we've done 50,000 terabytes of data. Every month, I end up buying about 12 terabytes of hard disk, just to process all this data. And we've only scratched the surface. You take the entire world's knowledge of the microbiome and put it together, we understand less than 10% of it. And that's only the gut microbiome. You add in more complexity of all the other microbiomes that's there in the body. You add the microbiome that's there in this room. I don't think we understand anything. And now there are companies that are trying to put probiotics into the AC duct. I have no idea whether that's going to be a good thing or a bad thing. Unilever released a product in Indonesia which is a probiotic wiping cloth. So you can clean your surface. The good thing is it's not antibiotic cleaning anymore, so you're not harmful chemicals are not being sprayed. But maybe it's a good thing to have probiotics cleaning your surface. Maybe it's not. We don't know. So there are so many different innovations that are coming out in this area, and I think we have to work a lot towards trying to understand what these microbes are trying to do. Just wanted to pull up one study that we did, and won't go into the details of the study except for this. We managed to, just in a few weeks of microbiome-based interventions, so this is nutrition and supplements, we managed to bring down the HbA1c levels. So if this can be done just by targeting the gut microbiome, I'm sure we can do therapeutics which can target the gut microbiome. There are companies that are, it's also believed that metformin, one of the most common anti-diabetic drug, also has an effect on the gut microbiome for the better. And that could be one of the mechanisms. There are institutes worldwide that are working on cancer therapies by targeting the gut microbiome. You name it, this microbiome-based therapeutics is probably going to revolutionize the way healthcare is researched. My guess is by 2030, 2035, around that time, both CE and FDA are going to get so many applications on targeting the microbes in our body rather than targeting the human receptors. The side effects are lesser, the efficacy could be better. Again, this is a graph from our reports. I mean, we've come up with a scoring system from -10 to +10. So again, I go back to the point of what is that super healthy microbiome that we're trying to target. So all the healthy that we see, all the average healthy that we see. So when I say average healthy, I mean people who've passed their blood test with nothing in bold or in the negative place. So they're between 4 to 6 is where we see the average healthy. But occasionally, once in about 250, 300 samples, we do get somebody who's going above the score of 6. And we're looking at the data, we don't understand the data. It seemingly doesn't look like such a super healthy gut, but all kinds of parameters are being thrown out to say that yes, this person seems to have a much better metabolic system within his body. So there's lots and lots of academic literature out there, and this also needs to continue to grow, which talks about the unhealthy microbiome, talks about the diabetes signature, the cancer signature, the depression signature. But we also need to start looking at what is the healthy signature. With all I'm from Bangalore. Every other building is a longevity startup, and every longevity startup wants to look into the gut microbiome and change it back to the microbiome which you had when you were a teenager. Is that the magic pill? Is that the magic solution? I don't know. I'm getting business, but. Science-wise, I don't know. This, I'm sure all of you all have seen this graph. The DNA sequencing technology every year is the price is falling down. In 2025, the price is even lower than what's there in the graph. But it's not. So when we launched the test in 2017-18, around that time, our retail price was 36,000 rupees. With great difficulty over the years, we managed to get it down to 10,000 rupee. my goal this year is to get the price down to 5,000 rupees to do one gut microbiome test. And if we can achieve that, I mean, it's not only people getting to know about their own gut microbiome, but researchers all around will be able to do many more samples to understand this. So that is one of the aims that we're trying. And the way we can bring down the price is not by, is one, of course, if we can crack the sequencing technology, great. I mean, if we can have an Indian sequencing company that can compete with ONT and Illumina, I'd love it. But it's all the peripheral solutions also. I mean, what are the enzymes that, what are the enzymes that are being used in the reagents that are being used? For example, we used to import, to collect the fecal samples, we used to import the tube from Germany. Till we found a good startup from ISE who could make that same solution at a better cost, and the quality was much better. Now, similarly, I've been throwing out challenges to different researchers in ISE saying that I want enzymes that can be made in India at a lower cost, but at the same or superior quality that we're getting from Europe. That is the only way that we can start bringing the price down. And with this, more and more research can happen. Before I end, it's not only wet lab experiments that we need to focus on. There's a lot of data that is there. I mean, there's a lot of public microbiome data that's out there, and with our expertise, here in India, in computational technology, can we have machine learning, simulations, AI, systems biology, all of that? Can we combine all of this and create in silico data for microbiome? Can we do drug discovery in silico and see how the microbiome reacts to this? This is also another avenue that we'll need to look at in terms of research, both in academia and in industry. My last slide. I mean, I hope and I wish we're not only the microbiome capital of the world, but we can become the biotech and synthetic biology capital of the world. We have the potential. We have the labs that are doing great research. We have companies that are doing really good products. And with a combined effort, I think we can really be the best globally. Thank you.
I request all the panelists also to join us on stage. Ms. Padmaja Ruparel of the Indian Angels Network. Dr. Debjani Paul of IIT Bombay. Ms. Malti Srinivasan of Pfizer Healthcare. Vasan Sambandhamurthy of Bugworks. We will introduce them at greater detail just before the panel. I request the speakers to stay on stage later for the Q&A.